For decades, the frontier of biology has been defined by tools like CRISPR, but editing an existing genome means working within the constraints of millions of years of evolutionary baggage. You are rewriting code you didn't construct in the first place. A different approach discards natural genomes entirely. Instead of editing, researchers are assembling life from scratch, using only non-living chemical components. The proof of concept for this is a synthetic entity known as SpudCell. It is built from about 150 to 200 distinct molecules, running on a minimal genome of exactly 90,000 base pairs, utilizing zero pre-existing cellular life. This marks a transition in the field. Biology is moving from a science of observation and genetic tweaking to a discipline of pure chemical engineering. In 1957, the launch of a simple metal sphere proved that space was an engineering domain rather than a distant mystery. Today, SpudCell serves as the biological equivalent. It demonstrates that basic cellular life does not require a mystical vital spark. It only requires the correct execution of highly specific chemical instructions. This assembly successfully executes three core biological functions. It metabolizes resources, it replicates its DNA, and it passes genetic information to new generations. By reducing these organic processes to programmable mechanics, scientists open the door to industrial-scale biological manufacturing. To understand how this works, we have to look at the blueprint. SpudCell operates under a strict limit of just 90,000 base pairs. This minimal genetic code is organized into exactly 7 circular structures, called plasmids. This allows for modular programming. Engineers can swap specific plasmids out to change a function without breaking the entire cellular system. Compare this to a naturally evolved eukaryotic genome, where instructions are tangled together in highly interdependent, overlapping networks. A minimal genome replaces that evolutionary complexity with predictable, executable logic. But this stripped-down code creates a biochemical problem. A 90,000 base genome is far too small to code for the complex internal machinery a cell needs to process energy. The solution relies on physical chemistry. The outer boundary of the cell is a liposome, a simple synthetic fatty vesicle. Instead of generating its own resources internally, the liposome feeds through physical fusion. It collides and merges with external donor droplets in its environment. During this fusion, the artificial cell absorbs vital components, specifically the enzymes and ribosomes required to read its synthetic DNA. In synthetic biology, complex metabolism can simply be outsourced to fluid dynamics, bypassing the need for internal biological complexity. A similar workaround applies to reproduction. Natural cellular mitosis uses complex protein spindles to pull chromosomes apart, pathways that SpudCell completely lacks. The mechanical alternative utilizes targeted accumulation. Specific synthesized proteins cluster heavily at opposing ends of the liposome's membrane. This localized clustering creates immense physical tension, forcing the liposome to mechanically pinch and snap apart into 2 daughter cells. Cellular division is ultimately solved as a basic physics problem, proving that biological replication can be engineered entirely without legacy systems. Researchers set up an in vitro experiment where variants of SpudCell were left to replicate and interact over the course of roughly 5 generations. This chart tracks the population dominance across those 5 generations. As the timeline progresses, a naturally occurring mutated variant line grows significantly faster and overtakes the baseline population. Darwinian evolution has been successfully triggered in a system constructed entirely from non-living chemicals. With the core mechanics now proving in a test tube, the focus shifts to industrial applications, known as cell factories. These programmable cells can be designed to manufacture targeted anti-cancer drugs, novel biofuels, and bioplastics at scale, driving a biomanufacturing market expected to reach $60 billion by 2030. The catalyst driving this rapid scaling is artificial intelligence. AI systems like AlphaFold are being used to generate novel protein structures that do not exist in nature. AI acts effectively as the compiler for synthetic biology, drastically accelerating the time it takes to write and test the code for these minimal genomes. Looking toward the 2030 to 2035 timeline, engineers are questioning the limits of the standard 4-letter DNA alphabet of A, C, T, and G. This brings us to xenobiology, the engineering of cells using artificial nucleic acids, or XNA. By utilizing entirely synthetic chemical bases, engineers can create organisms with alien functions, superior resistance to environmental stress, and tightly controlled built-in fail-safes. Biomanufacturing is moving toward operations built on an entirely synthetic chemical alphabet. However, the democratization of cheap, AI-driven synthetic design creates severe biosecurity vulnerabilities. Current global biosafety relies heavily on sequence-based screening. Security systems check submitted genetic code against databases of known legacy pathogens. The bottom-up architecture of systems like SpudCell proves that a malicious actor could use generative AI to design a highly virulent pathogen entirely from scratch. Because the code has no evolutionary history, it slips past sequence databases completely undetected. This necessitates an immediate global shift toward function-based screening. Security infrastructure must analyze what a sequence actually does, rather than just matching it against lists of what already exists. The underlying engineering logic of synthetic biology is now proven. Its long-term viability depends on our capacity to govern technology constructing life from scratch.